Well, I'd like first to thank the organizer to give me the opportunity to attend this very interesting meeting and to share with you some of our data on the influence of the intestinal microbiota on the gut adaptive immune system. So we have, as we have already discussed largely today, faced to the 14th bacteria in our intestines. The question is which forces control the host, the relationships between this microbiota and the host. So what we have learned is that we have been able across evolution to derive mutualistic relationships which are mainly probably largely based on the metabolic advantage that each partner takes from the associations, yet it remains that these bacteria are a major potential threat to the host, and against this the host has successively put successive layer of defense. First, innate and adaptive immune defense, which in vertebrate we have learned, especially from the work from Macpherson, that collaborate to maintain the epithelial barrier and protect local homeostasis and protect the host against systemic dissemination of these bacteria. So for today, I'm only going to discuss the adaptive immunity and trying to answer three questions. What is the nature of the intestinal adaptive immune response to the microbiota? Do we have any evidence that individual members of the microbiota have distinctive effects? And if so, what can be the mechanisms of these distinctive effects? So, as already alluded in the just previous talk, a very useful tool to address the question of the interactions between the microbiota and the host immune systems is to use gnotobiotic mice. And it's what we have done, and we have compared germ-free mice, conventional mice which have been colonized from birth by an SPF microbiota, as well as mice which have been colonized at the adult age, either with SPF microbiota that we call conventional mice, or mice colonized with selected bacterium or microbiota. And we have looked at the humoral and T-cell response by combining different techniques, classical techniques. So rapidly, a short word about the humoral response, which has been largely addressed in the previous talk. And indeed, in mice which are germ-free, there is no plasma cells in the intestines, and this the exposure to the microbiota results in the appearance of the IgA plasma cells, which secrete IgA, which can be found in the fecal, in the feces, and each bacterium that we have tested at a more or less level does induce an IgA response. And this IgA response is partly specific, as can be demonstrated by ELISpot if you look at the IgA response on lamina propria isolated cells. But besides inducing the appearance of a humoral response, what does the microbiota is to induce the appearance of T cells in the intestines. And so the question that we have had when we started this work was to know what was the nature of the T cell response to the microbiota. And in order to ask this question, what we decided to do as first is to use a relatively growth technique, which was to use to look at the transcriptome, at the mRNA expression at different level in the gut, and looking at the induction by comparing germ-free and conventional mice, comparing the induction of transcripts, both for innate response or for adaptive response. And as you can see here, as compared to germ-free mice, you can see, especially in the elum of the mice, a massive induction of transcripts encoding both innate response, specific of the epithelium, of the dendritic cells, of Th1 response, Th7 response, but also of regulatory response. And this is indeed due to the microbiota, because this can be reproduced if you colonize adult gnotobiotic mice with a mouse microbiota, and by day 20 there is already a very strong induction which stabilized and at the steady state being reached by 2 months. Well, looking at this, we had we were surprised to observe that the amount of transcripts was much higher in the ileum than in other parts of the intestine, and notably in the colon, despite the fact that the amount of bacterium is much higher here. And it was also very surprising because I have been actually trained by Delphine Guigrand, and for me it was obvious that if bacteria would induce a response, it would start in the Peyer's patcher or in the mesenteric lymph nodes, and then the response should be disseminated to the whole intestines. So some kind of response came from this work that we have already heard today, is that in the colon there is a very thick and solid layer of sterile mucus which could reduce the local stimulation of effector cells. So even if the cells have been stimulated and migrated here, they probably need to express their transcript to be restimulated. And here in the colon, it's likely possible in the ileum, because we have this fluid and discontinuous layer of mucus, but it's not possible in the colon. And to check that it was indeed the case, what we did was to isolate the lymphocytes from the different parts of the intestines. And as you can see here, and then we took these lymphocytes, and either by intracellular staining or by looking at their the release in the supernagens after stimulations, you can see that indeed it's the same exactly in the ileum, in the colon, in the duodenum. You induce, as compared to germ-free mice, T cells that you can isolate from mice which have been colonized with a complete mouse microbiota, have strong Th1, Th17, but also regulatory response, and also a Th2 response. And I should say that you also see an increase in the number of FOXP3 positive cells. Perhaps one word is that if you look by intracellular staining, at least in our germ-free mice, the few T cells that you find in the gut can produce interferon gamma, can produce IL-10, and you have a few FOXP3 cells. But there is one thing which doesn't exist in the germ-free mouse. It's really the IL-17 response. So clearly, mouse colonization induce a complete mouse colonization by a complete mouse flora, induce a large spectrum of immune response which balance each other, including secretory IgA, innate gene, at least in the ileum, and a spectrum of Th1, Th2, Th17, and Treg response, which which again balance each other and maintain the homeostasis in the tissue. A first question which arose from this observation is, and which was alluded already for the IgA, is the question of the specificity of this response. And to address these questions, I have taken advantage of a work that we have been associated with, but which has been, in fact, done in the laboratory of Gérard Héberle, which has consisted to use mice which have a single transgenic T cell receptors, which in this case was directed against the male antigens and which is unable to react with microbial antigens. Gérard was mainly interested to look at the fate of cells which express the nuclear factor Ror gamma that he follows by using GFP. Ror gamma, which can be expressed in both regulatory and Th17 cells. So it's why he focused on this population. And whereas in control, mice, which have diverse T cell receptors, you can see both FOXP3 and Th17 response. What is interesting is that in these mice, which have a single transgenic receptors, in fact, you have no FOXP3 positive cells, indicating that you need to recognize the antigens to be able to drive regulatory T cells. In contrast, you have a nice expansion of lamina propria Th17 cells. This is not only true in these mice with a T cell receptor which recognize the male antigens, but he confirmed it in mice with a distinct TCR against egg lysozymes. And this is indeed related to stimulation by the microbiota, because it can be removed in the presence of antibiotics. So, in fact, when you see that, you wonder whether the Th17 response is really a true adaptive response, or maybe in between innate and adaptive immunity, at least part of the Th17 response. So, at least for the first question, I think that our results, as well as others, show that there is indeed a large spectrum of response which are induced in response to the complete microbiota. in the normal state, in an immunocompetent animal, these responses balance each other's, but some of which may occur independently of specific antigen recognitions. So the next question is, do we have evidence that individual members of the microbiota have different effects? And what can be the mechanism? And what I would like to try to take you is that in fact these distinct mechanisms depends on the trade-off that the host and the bacteria will take together depending on their individual fixtures, both on the bacterium and the host side. So the first evidence that microbiota may have distinctive effect on the immune response came from global transcriptomic studies that we did by colonizing mouse intestines with a whole human microbiota that we had taken with the idea that we will find the bugs which were interesting directly in the human microbiota. And to our surprise, here in gray you have, and white you have conventional and conventionalized mice, and here the intensity of the immune response compared to the metabolic response and the cell cycle response. And you see here that, to our surprise, the human microbiota induced very little immune response, while it induced very nicely a metabolic and a cell cycle response, at least in the ileum of the mouse. So we did a very large number of colonization by individual bacterium, by more complex flora, including the Schedler flora and this whole human microbiota. And in fact, by this relatively gross technique consisting in looking for mRNA encoding regulatory and pro-inflammatory genes in the ileum of our gnotobiotic mice, we had no detectable response. So we decided to come back to the mouse flora and to use a culturable fraction of the mouse flora. And I should say that in that case, we had indeed a weak and inconstant induction of mRNA encoding mainly FOXP3 or IL-10, so a regulatory response. But I think you have to remember that it's relatively gross, and I think we have now, from a number of papers, good evidence that many microbiota members, either individually or collectively, can induce regulatory response. And all these papers, and the last one being one that we just heard from Andrew McPherson, which has just been published. And so if I want to summarize what we can imagine from this paper, what I would say is that probably for most of the members of the microbiota, the trade-off with the host is probably, and here I've taken the title of this paper, which for me was one of the most illuminating paper in this area, which is that the bacteria are able to simultaneously induce IgA, and this IgA can probably keep the bacteria in the lumen, in the mucus, while limiting potentially immunostimulatory contact with the host. And at the same time, these bacteria can induce regulatory T cells, which avoid both innate and adaptive immune response, which can be potentially deleterious, not only for the host, but also for the bacteria. And what is interesting is that, in fact, this regulatory response can probably promote the IgA response, because it has been shown, both in this paper and by Sidonia Fagarazan, that regulatory T cells can give rise to follicular helper T cells. Yet, even so, probably most of the microbiota is able to induce a regulatory response. At least the whole microbiota induced an inflammatory response. So we really needed to understand what was going on. And in fact, as you can see, we are stricken by the fact that the whole human microbiota didn't do much, and that the culturable fraction of the mouse flora didn't do much either. So we reasoned that we needed something which was from the mouse and which was in a non-culturable fraction. I should say that by looking at by fish analysis, we had in the feces of our colonized mice a small decrease of some Clostridium groups. So we decided to enrich for Clostridium by taking the sporulated fraction of the mouse flora, which can be done by heating the mouse flora. And indeed, when we colonized our mouse, we recapitulated all the pro-inflammatory and regulatory effects of the microbiota. So this could indicate that either we needed a complex microbiota, but in fact, and we have already heard a lot about these bugs, there is a bug which has been described 30 years ago, which was an attractive candidate because it had been shown in the past as a very strong inducer of IgA response and also an inducer of the recruitment and activation of intraepithelial lymphocytes, which is actually, which responded to the criteria that we had for our pro-inflammatory, well, our immunostimulatory microbiota. It was Clostridium related, spore forming, non-culturable, interestingly conserved during evolution. Even more interestingly, settling in the rodent intestine at time of weaning, which is just at the time where the maturation of the gut adaptive immune system starts, and with host-specific adherence to the ileum, meaning that the mouse SFB cannot adhere to the rat ileum and conversely. And this bacterium had been purified by passage in germ-free mice. So Hans Snell, who had spent his PhD doing this, gave us the strain, and so we could put it in mice. And in fact, what we could done is more or less recapitulate, perhaps not as efficiently as the whole sporulated fractions, but quite efficiently recapitulate all the T-cell helper T-cell response that we have seen. I've only shown here the response of the cytokine, and here it's conventional microbiota, the SFB, as compared to the culturable fraction of the microbiota. So if you look at the IL-17 response, the culturable fraction of the microbiota does induce a little bit of it, but clearly this is a log scale. SFB is much more efficient, and SFB alone is able to recapitulate most of what does the conventional microbiota. So it seems that SFB can be considered as the key driver of the gut immune response, being able to recapitulate the innate gut immune response. I've not shown the results, but it's induced strongly, and we've already heard about that, the microbicid barrier. It's especially a strong inducer of REX3 gamma and MMP7. It's induced at SHGA and CD8 and CD4 response. So what are the consequences for the host of this induction of the gut immune response? At least it doesn't do any pathology because the intestine of the host is totally healthy. However, what does SFB is, in fact, to help the host building a barrier effect, and this has been nicely shown by the work of Ivanov and Dan Littman, published at the same time. We show that SFB was able to confer a barrier effect against the invasive pathogens we heard this morning, Cerodentium. And I think the fact that this induction of intestinal T-cell response is probably very important as a safeguard in the intestines. If the bugs, the commensal bugs, have been able, in fact, to escape the epithelial barrier the defensin, and the secretory IgA, here there is a place which can help them, which can help the host to get rid of them. And as an example, I've shown you here what happens in the patients with HIV infections at the stage of AIDS, where there is no more CD4 T cells in the lamina propria. So this person has no more gamma interferon to arms the mucosal macrophages, and these mucosal macrophages are unable to eliminate atypical mycobacteria, which normally should be eliminated by the microbiota. So I think that this barrier is absolutely key. So the question, and since Andrew Macpherson published last week a paper where he suggested that SFB is not a true commensal, I decided to try to answer the questions. And I think somehow it's true that it's a bizarre commensal because most of the commensals are probably in the mucus, as we have already heard. But this bacterium is very specific because it is able to attach very strongly to the epithelium. And in fact, as you can see here, maybe it starts to divide when it attaches to the epithelium. So it looks a little bit, this adhesion behavior looks like a little bit like a pathogen. Yet you have to remember that, at least on the basis of the morphology, these bugs has been found in many species across evolution. There is apparently related 16S RNA sequence in different species. So it seems that it's really some kind of specific symbionts which likes to be in the intestine. So what I would like to show you is that, in fact, try using a work we have been doing recently to try to explain what I see as a commensal. And I think a commensal is a bug which doesn't induce any pathology in an immunocompetent host and which is able to settle in the intestine as a result from a trade-off between nutritional benefit and resistance to stress in the host gut. And to illustrate that, I've taken the example of this work that we've been doing using E. coli K12. You have to remember that E. coli K12, which was isolated years ago from the feces of somebody, is no more really adapted to the intestine, but it's more a lab strain. So if you put it in a gnotobiotic mice, in fact, you induce very rapidly mutative adaptations. And there are mutative adaptations because if you take the facet of the mice and put them back in a gnotobiotic mice, then you don't see any more mutative adaptations. So that was been work done by Antoine Giraud and François Tadei a long time ago. And we wanted to understand what was exactly these mutative adaptations. And what we observed is that, in fact, very early on, when you put the E. coli in the gut, we see the very rapid emergence within 2 days of OMZ mutants, which actually have become highly resistant to biliary salts. And this is because they have switched their large pore to a small pores, but at the same time, eventually they cannot take as much glucose as the others. So it's okay at the very beginning of the colonization when there is not so many bugs in the intestines, but when they reach a high density, in fact, their growth is not so good in low nutrients, low nutrients in case of nutrient restrictions. And in that case, you see the progressive emergence of 2 different mutants, which actually are able to rapidly grow under nutrient restrictions at high bacterial density. So it seems that, at least for these bugs, which live in the mucus, to be able to live in the mucus and in the gut results from a trade-off between bile resistance and nutritional competence. And perhaps just a word for immunologists is that these adaptations, these 2 mutants here, have lost the flagellin. So an obvious question was whether TLR would be important to drive the diversification of the mutants. But to our deceptions, the immune system has nothing to do with that. First of all, we have exactly the same diversification if we take MYD88 mutants. Secondly, we can reproduce exactly the same things in a chemostate, where there is no immune system. So, but perhaps we have to remember that bile salts are probably the first defensins and finally act as defensins. So at least in the lumen, probably biliary salts should be taken into account perhaps as some kind of immune defense. So to come back to SFB can be, has some kind of pathogenic-like behavior because it strongly adheres to the mucosa, and probably this results in a very strong innate response, which is necessary to drive the strong adaptive response that we see. But probably it's my guess is that our guess, in fact, and it's a work that we are pursuing with Pamela Schnupf and Philippe Sansonetti, is that perhaps these bugs need to attach to the intestines to start dividing, and it's probably why he has started to have this type of trade-off coming close to the intestines. And the induction of the host inflammatory response finally may not only be, obligatory, but perhaps may be useful to eliminate competitors and gain access to the ileal mucosa at the time of weaning, something which has been described for Salmonella. And subsequently, the host-induced IgT cell response may be useful to limit SFB and maintain homeostatic relationships with the host, but somehow it may also be useful for SFB to keep its niche free from intruders. So So if I want to come back and to come back to the possible trade-off that we have between different members of the microbiota, I would suggest that for most of the bacteria, the bulk of the bacteria which thrive in the mucus, probably the best trade-off is to result in for the host to induce IgA and regulatory T cells. For bacteria needing a more intimate relationships with the host, probably there is no way the homeostasis is maintained by inducing a different balance between pro-inflammatory and regulatory T cells, and it's possible that this type of bacteria, SFB being the prototype of such bacterium, but one can imagine that some other bugs, for example, like Helicobacter pylori and so on, may have perhaps play a specific role during evolution in shaping a local host defense. Yet, does it mean that these bugs do not induce any pro-inflammatory T cell response? But we heard already what the response, the answer from Andrew McPherson's, and I would like in my last slides to take this as an example that the outcome of the host immune response really depends on the host factor and the host immune systems. Taking, for example, this E. coli, which is found in the normal healthy gut, but it's largely enriched in the ileum of Crohn's disease patients where it becomes enteroinvasive. If you take these bugs and you put it in the gnotobiotic immunocompetent healthy mice, there is absolutely no evidence of induction of pro-inflammatory response, either gamma interferon, TNF-alpha, IL-1 beta, or IL-17, and these bugs behave exactly like a K12. The situation is completely different if the mice express a receptor for the pili of this LF82, and this has been nicely shown by Frédéric Carvalho, actually the guy that was called this morning La Machine. And when he did his doctorate in Clermont-Ferrand, he did this nice work. And so what happened is that it has been shown that in the ileum of the patients, SECAM appeared as a consequence, in fact, of the local production of inflammatory cytokines in genetically predisposed individuals. And here what they have done is to use mice which express, we have a transgenic expression of SECAM in the colon, and in that case, the bugs becomes extremely pro-inflammatory. So I think it's very important to remember that even an innocuous bacterium can become a dangerous foe in predisposed or modified hosts, and it's really the trade-off between the host immune system and the bacterial features which decide the outcome of the response. So to conclude, in a healthy gut environment, the microbiota, both the peacekeeping bacterium and probably some bacterium which have been coined under the term of pathobionts, and probably SFB could be put as a pathobionт, results in physiological inflammation, which contribute to the barrier effects against the microbiota, protecting the host against pathogens, but also protecting probably the niche of the bacterium. The situation either can change if a factor in the host environment changes, either because the host has a problem with his immune system. But if you change, and we have already heard today many evidence of this, when you change the gut environment by putting antibiotics, by changing the diet, you can change the ratio between this peacekeeping bacterium of these pathobionts. You can probably, this is probably very important, allow this even peacekeeping bacteria to become adherent, and then you change the ratio and you induce inflammation. And this is probably important to take into account for therapeutic intervention. And then I would like just to finish by thanking the people who have participated in this work, and particularly Valérie Gaborio-Routiot, who is the person in the lab, who is the senior researcher in the lab, who does a lot of work, who has been, I couldn't have done this work without expertise in gnotobiotic mice with a number of talented postdoc or PhD students. We have had a very nice collaboration with Gérard Heberl and I also want to thank Hans Snell who gave us the bacterium. Denise Kelly, Ilmke Mulder, and Aiklan, who have helped us with transcriptomic studies. This work done with Antoine Giraud and François Tadei, who are evolutionists and who have taught us a lot about the interactions between the host and the microbiota, and recent work with Philippe Sansonetti and Pamela Schnepp.